Hello, my name is Joanne Nazario, and we're going to be talking about the formation and structure of cytoskeleton fibers. Today we're going to be discussing the topics of cytoskeleton. We're going to discuss the three types of cytoskeleton fibers. We're going to discuss them individually: microtubules, microfilaments, and intermediate filaments. What is the cytoskeleton? The cytoskeleton gives cell the ability to change their shape, regulate locations, coordinate movements, and interact with their environment. The cytoskeleton is known as a meshwork of protein filaments that are found throughout the entirety of the cytoplasm. The cytoskeleton is constantly changing in response to signals. This structure is known as the bones and muscles of the cells and allow for all cellular mobility within the cell. Cells are ATP-making factories and are highly organized and compartmentalized, all allowed by the cytoskeleton. One of the most important processes that utilize the cytoskeleton is mitosis and cell division. There are three types of protein filaments that compose the cytoskeleton. There are microtubules, microfilaments, and intermediate filaments. Each are arranged and formed in their own way, and all have their own various functions. They are structurally and functionally different. Intermediate filaments are composed of various proteins of a given subunit. Microtubules are composed of many tubulin proteins. The microfilaments are made of actin proteins. All of these have thousands of proteins that come together to make long fibers out of protein complexes. They intertwine and bundle to create the meshwork. Microtubules are hollow tubes that can rapidly dissociate at one location and reassemble elsewhere. The centromere plays a huge role in the creation of microtubules. The centromere is the point of origin for microtubule production, and they extend out, and the microtubules extend out of the centrosome to the cell membrane. This then allows the production of tracks and allowing walking proteins to move cargo around the cell. Microtubules are disassembled and then reassembled into new microtubules when needed. Microtubules are made up of tubulin, which are dimers of two globular proteins. These proteins contain one alpha and one beta subunit. Tubulin has two ends that are physically and chemically different and allow proteins to know which direction to move. The negative end will be embedded in the centrosome. The positive end will point out and grow outwards towards the cell membrane. Microtubules can disintegrate without a warning. Tubulin dimers can be bound to GTP, allowing for higher dimer affinity, or they can cleave GTP to GDP, allowing for less affinity. A GTP cap can allow for the overall stability when hydrolyzing dimers. These dimers are ultimately unstable because of the simultaneous state of dimer hydrolyzation and dimer building. This is known as dynamic instability. Intermediate filaments are specialized by their strength. They allow for the cell to withstand stress such as stretching, pulling, and squeezing. Most of the time, the intermediate filaments are found in the cytoplasm where they line and attach the nucleus all the way out of the membrane. The nuclear lamina is an underlying complex that acts as a nuclear cortex. Intermediate filaments are made up of individual fibers that are twisted into strands. Two intermediate subunit proteins will wrap around one another to form dimers, and those are staggered to make a tetramer. There are four main classes of intermediate filaments. Keratins are the broadest type of IFs found in the cytoplasm, and are diverse because each keratin is made up of a combination of various subunits. Vimentin is found in areas that must be durable and stretchy and tolerant to mechanical stress. These are found within connective tissues, muscle tissues, and cells of the nervous system. Neurofilaments provide most of the structural support of neurons, especially the axons. Nuclear lamins are for the stretching of the nuclear envelope. Microfilaments are made of actin subunits. These actin subunits will align in a double helix form to create filaments.